Hello, and welcome to another how-to video from Ronawk. In this video, we'll quickly review the process for seeding E-blocks with cells. Note that this video is a supplement to the Ronawk E-block protocol. Start by gathering the items you'll need. These include E-blocks, sterile pipette tips, a growth vessel, where we're using a 6-well plate in this video, sterile Hanks balanced salt solution without calcium and magnesium, sterile growth media, where you'll need around 8 to 10 mils for each block you're seeding if you're using 6-well plates. And finally, your starter culture. For best results, you'll want your starter culture at a concentration of 2 times 10 to the 5th to 2 times 10 to the 6th cells per mil, and you'll need around a half a mil of starter culture for each block you'll be seeding. Step 1 is the initial seeding of your block with the starter culture. Start by removing your E-block from their packaging. Note that the top of the block is indicated by a dot on the packaging that is labeled top. Using a sterile spatula, place each E-block into your growth vessel with the top side facing up. Next, use a sterile pipette to remove any excess HBSS from your growth vessel containing the E-block. Now, use a sterile pipette to add modified HBSS to each growth vessel until the E-blocks are submerged. Let your E-blocks equilibrate in HBSS for at least 30 minutes. Remove the modified HBSS solution from your growth vessel using a sterile pipette. Now gently add a half a mil of your starter culture to the top of each block. Do this in a dropwise circular fashion as shown here. Now wait 2 minutes. After 2 minutes, collect the excess starter culture that has not been retained in the block and reapply this to the block dropwise as you did in the previous step. Again, wait 2 minutes. Repeat this collection and reapplication 1 to 3 more times, depending on the cell type and block coding. Step 2 is the addition of sterile media. Gradually add sterile media to the far edge of each plate well. In other words, add the media to the well and not directly to the block. Continue to add sterile media to each well until the block is fully submerged. This is typically about 8 mils per well. You're now ready to place your plate into the incubator and grow your cells. Thank you for watching, and we hope this video was helpful. Check out our YouTube channel for additional how-to videos from Ronawk, and use the Contact Us button on Ronawk.com if you have remaining questions.